Hey guys, welcome to today's video lesson. We are talking about exchange of nutrients and wastes. So the purpose of today's lesson is to look at characteristics of absorptive surfaces within the digestive system, specifically focusing on the structure and function of the villi. We will look at digestive enzymes, their role in chemical digestion, that being amylase, protease, and lipase mainly. Then we'll also look at the excretory system, that being the kidneys. Kidneys are responsible for filtering blood to remove nitrogenous waste. That's a byproduct of cell metabolism or cellular respiration. So we'll look at the different types of nitrogenous waste depending on what type of organism it is. We will also look at, in mammals, the function of the kidneys, and the kidneys contain these little structures called nephrons, and they are responsible for filtering our blood. So we'll look at each section of the nephron and what happens at each stage. And then we'll just, again, talk about that a little more specifically. So you can write a condensed version of the learning goals here. You can pause the video. Okay. So let's talk the absorption and digestion of food. So this is from chapter 7 of your text. And I'll start off here. So we're looking at the digestive system, and basically this is the process of ingesting food, absorbing all the nutrients we need from it, and then eliminating the waste from our body. This process starts in the mouth. So here we ingest the food, and there's two types of digestion. Digestion is simply the process of breaking down food into its smallest component. There's 4 major components, so that it can then be absorbed into the body, and then what's left is just the waste that is excreted. So of our food, there's 2 types of digestion. There's mechanical, that is the physical breakdown of the food, and that starts in the mouth with the teeth. But there's also other examples of physical digestion. It occurs in our stomach. Our stomach is muscular, and it churns, it pulls and crushes and contracts on the food, kind of tugging it and pulling it apart. But then we also have in the intestines this movement, this contraction movement that moves food through the intestines. It's called peristalsis. But that starts in the mouth, mechanical digestion, but chemical digestion also takes place in the mouth, and that is we have saliva, and within saliva there is an enzyme called amylase, and amylase works to immediately begin breaking down carbohydrates, that's being sugars, into their simplest form. Anyways, we have saliva glands that produce saliva to do that. It can either go 2 ways when we swallow down our air pipe, the trachea, or the food pipe, the esophagus, but we have this flap called the epiglottis, which closes the air pipe when we swallow, so it goes down the esophagus into the stomach. It can be there for minutes or hours, where it is then further digested mechanically and chemically. We have hydrochloric acid, which is produced by these cells in our stomach, and hydrochloric acid has a pH of about 1, so it chemically breaks down that food as well. From there, we have other organs, including the liver, the gallbladder, and the pancreas, and they secrete further enzymes which break down the food. So proteases, as it suggests with a P, they work to break down proteins into their simplest form. We also have lipases, the enzyme which acts upon lipids, that being fats, to break them down. It's no longer food, it's food before it's ingested, but once it's ingested, it's actually called chyme, that's C-H-Y-M-E. That chyme passes through the small intestines. We'll talk about that more in a sec, where it then goes into the large intestines, and by this stage it's just fecal waste matter, which is then deposited through the rectum and out through the anus. So technically, the tube within our body, if I had a donut. I'll show you a donut, and there's a hole within the donut, and you can put your finger through the hole of the donut, but technically your finger isn't in the donut. It's still outside of it, even though it's inside. That's like this tube, this big, long, single tube that comes all the way through our body to the outside. If something is in this tract, it's actually not yet in our body. It needs to be absorbed out of that tract into our body. And so there's a few features that allow this to happen. A key stage of absorption is here in the small intestine. So here it's been broken down and broken down. When it gets to the small intestine, there's a lot of fluid to the chyme that we want to absorb out of that, what will be left as waste matter, into our body, into our blood. And so water absorption takes place in the small intestine. But just as, or more importantly, is this is where a lot of our nutrients are absorbed. And our nutrients are absorbed through the structure of what we call villi, or singular being villus. And what they do is they line the inside of our small intestines, and they have all these villi are finger-like projections that line the inside of these tubes. And if we zoomed in on this finger-like projection, this villi, you would see that even these fingers have little fingers called microvilli, and what they do is they act to increase surface area. So we want to absorb as much nutrient out of that food before it's excreted from the body as we can. So villi are so important in that, to the point where people who have trouble with their villi, they have difficulty absorbing nutrients, gut issues, and you've probably heard of the term celiac. It refers to this, and here's a microscopic image of villi. You can see these finger-like projections, and microvilli would be on them as well. But what they do is they increase surface area to absorb nutrients, but for diffusion or for something to cross a membrane, we also know that it has to be moist, and that's our digestive tract. It's very moist. It's inside our body. These surfaces are very thin. It's just like a single cell up to 2 cells thick, and large surface area. We said villi does that, and there's also a concentration difference. So within the intestine, there's high amounts of sugars and proteins and fats and all of that. But in our blood, there's a lower concentration. So through passive but also active transport, those nutrients, say if this is the tube, this is the villi in the tube, those nutrients diffuse, are pumped out of the digestive tract and now into the body. And there's 2 modes that it can be absorbed now, or 2 modes of transport that those nutrients can be extracted out of the chyme into our body through. First up, we have these blood vessels, this capillary network, so that those nutrients are absorbed into our blood and transported throughout the body to every single cell needs energy. But then we also have what's called the lymph system, or the lacteal duct, and we can get nutrients into the lymph system, and it then goes to the liver, and the liver will decide what has to be done with those nutrients from there. Where should they go? What should they be built up into? So that's what happens in the small intestine. It is then passed on to the large intestine, where it is excreted. And so the learning goal was to talk about absorptive surfaces, how structure and function of the villi allow for digestion to take place. And we've said these things. We've said that there's 4 main types of macromolecules. If food were broken down into its smallest parts, it's broken down so they can then be built up into what we need it to be. There's a few enzymes that work to chemically break down the food as well. We have amylase in the mouth and the saliva. But then we also have proteases, which break down proteins, and lipases, which break down lipids, and they're secreted by our other organs such as the pancreas and the liver and the gallbladder, so that by the time it gets to the small intestines, it's really broken down and now it can be absorbed through the villi and the villus and the microvilli. There are these finger-like projections that do the hard, heavy lifting of digestion, that is breaking food down so that it can then be absorbed. That is the digestive system. And now if we move on, We can talk about, well, that's not the only system in our body that is really helpful in excreting waste product. As a byproduct of cellular respiration, that is metabolism, taking in oxygen and taking in sugar to create energy, water isn't the only, and CO2 only byproducts. There's this nitrogenous waste. That's this amine group with a nitrogen part to it. And it's extremely toxic. If it builds up, then it's fatal. And so different organisms have different ways of excreting that waste. And in most aquatic animals, they convert that into ammonia. It's less toxic. And then we, mammals, convert it into what's called urea. But insects convert it into what's called uric acid. It's kind of this whitish paste. Urea needs to be diluted. That's why when we excrete our nitrogenous waste, it's diluted, but with lots of water, it's this fluid. Fish don't need to dilute it much because they're living in water. They can just excrete it in a more concentrated form. So if we talk about how do we as mammals filter our blood to get out those nitrogenous wastes so that we don't toxify and poison ourselves, well, that's what our kidneys are for. And our kidneys have this really big supply of blood. So if I had an image, I would show you a picture of our heart, and from our heart comes the aorta, and then it branches, it goes up to supply like our arms and our head, but then it also comes down, and that's called the descending abdominal aorta, and from there it branches and it splits, and we have two kidneys about the size of our fist halfway down our back either side of the spine. They get this big blood supply, and they filter our blood to clean it out. So there's heaps of blood that pumps through these kidneys every second, our whole life. People with our kidneys, that's called renal failure. Renal means kidney. They die if they're not hooked up to a dialysis machine. That means they go into hospital for hours every week, or spend tens of thousands of dollars and get one of these machines at home. So what our kidneys are doing is so important, even though we don't often think of what they're doing. But if we zoom in on the kidneys and talk, all right, how do they actually filter our blood? Well, they have this functional little unit called the nephron, and our kidneys are made up of millions of nephrons, all doing the same thing. And what happens is we have this blood supply, this renal artery, and it branches and branches and branches until it gets to the smallest blood vessel unit, that's the capillary. And from there, the capillary meets the nephron, and the nephron is this kidney structure that's responsible for taking the waste out of our blood and then excreting it from our body in the form of urine. So let's zoom in now on the nephron. We have, here is a capillary. And this capillary forms this ball of blood vessels, and we call that the glomerulus. The glomerulus is surrounded by this capsule. This is where the nephron begins. This capsule is called the Bowman's capsule. And this is where the blood is filtered. So high pressure blood is pumping in here. And a lot of the contents of the blood is just forced. It's pushed out of the blood and out into this nephron. So even though there's a lot of pressure, big things like blood vessels, platelets, they don't move out of the blood, but a lot of the rest of it does. So that's like our water, our salts, that's sodium and chloride and other ions such as potassium, even sugars, as well as the nitrogenous waste is pushed out of our blood and into the nephron. So the rest of this process is all about keeping that waste out of our blood so that it can be excreted, but also reabsorbing, keyword, the good things that we want in our blood back into the blood from the nephron. Those good things are water and salts and sugars. And so what happens now is just these series of steps to reabsorb good things. And so we have a few parts of the nephron. We have the proximal convoluted, that means like confusing, squiggly tubule. Proximal means close. And here we have a few more good things that are reabsorbed. And then we go go across, and we go down to the loop of Henle. This descending loop of Henle is where we reabsorb most of our water back into our blood. You can see that capillaries surround the nephron. Then as we go up, this is where a lot of the salts are reabsorbed. That's sodium and chloride. And then that continues in the distal convoluted tubule. Then we get to what's called the collecting duct, and that's where other nephrons join up and empty the waste product. And this is just one last chance to reabsorb the good things from a nephron back into the blood before it's excreted, but also, keyword, to secrete any final waste products we want to get out of our blood back into the nephron. And so that is the process, and we've spoken about a few of these features. Selective reabsorption, that means how we can like selectively choose what comes back into our blood after it's all been pushed out, and that happens through a process of passive transport but also active transport. And we've said secretion, that is one last step to remove the wastes. But I'm going to take us to a summary slide just to look at that. And here we go again. So we have the glomerulus and Bowman's capsule here, and this is where the filtrate, that means like what's been filtered, comes out of the blood and into the nephron. It's composed of water and ions, sodium, potassium, chloride, and other small molecules such as sugars and salts and that. But in the proximal convoluted tubule, we reabsorb some of those good things, including glucose, amino acids, calcium, good things. We want to get rid of some of these waste products. You can just know them as nitrogenous wastes. The loop of Henle, descending limb, is where we reabsorb a lot of that water. And then the ascending limb of the loop of Henle is where we reabsorb that sodium and chloride. Remember, that's NaCl, that's salt. We need that in our body. And then we just continue to do that in the distal convoluted tubule. And the collecting duct is that last chance to reabsorb the good things, last chance to also secrete the waste products that we want to get out. That's ammonium and hydrogen ions. But you can know them again as nitrogenous wastes. And that, if I showed you another image, this is all these tubes, they collect up to what is called the ureter. And the ureter, that's it here, joins up to the bladder. So we've got two kidneys, two ureters, and from the bladder there's a single tube called the urethra, and that gets the waste product now fully filtered and diluted, and now it's urine to the outside world. And that is how we filter our blood to excrete nitrogenous wastes. So, your homework, you may recognize this. It looks a little bit like a nephron, the yellow being the nephron itself, and the capillaries in there somewhere. That looks like a glomerulus. But your homework is to firstly read chapter 7, add some keywords into your book, definitions, and then to create something that represents a nephron. That may be virtual, physical, tasty, or it can be something different. But don't just create it and not know what's going on. Make sure you can describe what's happening at each step. How does structure relate to function? And that is all for today, guys. I hope that this video has been informative, and maybe later down the track leading up to your final exam, you'll be able to watch over this and just let this remind you of what we have covered so far this week. But God bless you. I'll see you soon.